Hello, and welcome to the iScience video on research into cell cycle checkpoints. Cells divide to form new cells during a process called the cell cycle. At different stages in the cell cycle, the process stops so the DNA of the cell can be checked and repaired. These checkpoints are being investigated by Professor Peter Burgers from Washington University, who recently came to the UK as part of collaborative work with Imperial's own Professor Xiaodong Zhang. While here, he lectured on his work and started by explaining cell cycle checkpoints.
My lab works on DNA replication and DNA damage responses and DNA repair. I'll be solely talking about one aspect of what my lab works on, and that's DNA damage checkpoints. At each point during the cell cycle, the CDK cyclins that drive the cell cycle can be inhibited by checkpoints. And so these can occur at the G1S interface of the cell cycle. They can occur during the S phase of the cell cycle, and they occur at the G2M phase of the cell cycle before mitosis.
It's already known that these checkpoints in the cell cycle are controlled by a regulatory protein kinase called MEK1, but the work of Professor Burgers has identified 3 activator proteins for MEK1. One of these is called 911, and I asked him what the 911 protein was used for.
There are several processes it's used for. So the one process I identified was checkpoint activity of 911, where it loads onto DNA at certain regions, at certain junctions of DNA in order to mediate checkpoint activation of MEK1.
To activate MEK1, the 911 protein needs to be loaded onto the DNA. Again, Professor Burgers explains.
For loading, there's a set of what's called clamp loaders in eukaryotic DNA metabolism. For the checkpoint we're really interested in, 4 of the subunits remain the same, but there's an extra 5th subunit, which is called Rats-van yeast, known to be involved in checkpoints as well, and it will load 911, and then there are 2 more branches that are involved.
Once the 911 is loaded, it can activate MEK1 to repair the damaged DNA. Although 911 is not the only protein that activates MEK1 for DNA repair, it is one of the most important. It's involved in the G1 checkpoint of the cell cycle, the G2 checkpoint, and the S-phase checkpoint. The next question was the tantalizing one. Does the research apply to humans?
What we find in yeast is directly applicable to what happens in human cells. I mentioned the situation where we identified key amino acids in 911 that involved in checkpoint activation, and one of these key amino acids in the analogous human gene is actually involved in mutations in that amino acids are involved in adenocarcinoma in human patients. So there's a direct applicability.
Finally, I asked what may happen for the research in the future.
What we don't understand is how these enormously big proteins, how they actually work. So what we need to do, and that's why I'm here and collaborating with Xiaodong Zhang, is we need to basically go to the structural studies, carry out structural studies in order to see what these proteins look like, where some of their cofactors bind and how they work, and then go back to the biochemistry and back to the genetics and then back to the structure again. Kind of walk around this circle between structure, biochemistry, and genetics in order to understand how this really works. At the moment, all we know is that it works and that this is required and that's required, but we really have no idea how it works.